Right, good afternoon, everyone, and thank you so much for joining us. We're really looking forward to hearing Daniel's presentation this afternoon, and welcome, Daniel, and congratulations once again. Daniel is our bronze medal winner for 2022 for his PhD thesis on Thesium. So really looking forward to that. And what I'll do is I'll hand over now to Anina, who is going to formally introduce Daniel, and then he'll get on with his presentation. Thanks, Anina.
Great, thank you very much, Glynis. Welcome, Daniel. We're so glad to have you here today. And welcome to the audience as well. It's great that you can all join us from the different corners of South Africa. So we're very grateful that you're all here, and we're looking forward to this. Just as a quick introduction, where Daniel has come from and what his career path has been so far. He started studying in Nigeria, where he did his BSc honors in botany at the University of Maiduguri in Nigeria, and thereafter he did his MSc in plant biology at the University of Ilorin in Nigeria. And from there on, he made a move to South Africa and Cape Town, where he did his PhD at the University of Cape Town. And this was supervised by Professors Muyase and Verboom. And that's, of course, the main topic that we'll be hearing about today. And then after that, in 2021, Daniel did a postdoc at UCT under the very prestigious Smith's Memorial Botanical Memorial Fellowship, which is convened by UCT and SAAB. And during that time, he studied the phylogenetic community diversity of Southern African quartz field habitats, which I'm sure was also very interesting. So currently he is a lecturer at Gombe State University in Nigeria, and there he teaches taxonomy, biosystematics, and evolution courses. And I'm sure he's making a very valuable contribution there, since there's just a handful of evolutionary biologists and plant phylogeneticists in that area in the West Africa. So we're very pleased that you can share your knowledge and skills back in Nigeria. And we're also very grateful that you can present to us today. Thank you so much for the time you put into this. Daniel is presenting live from Nigeria today. So of course, as we know with webinars, there might be some hiccups or glitches. So if there is any lagging or problems, please just be patient, see if it gets better by itself. And if not, then you can just leave a note in the chat and we'll see what we can do. But most of the time, it's just an internet problem on someone's side that we just need to wait for it to sort out by itself. But otherwise, I'm sure everything will run smoothly. Just for all the listeners in the audience, please remember to keep your microphones and your cameras off during the talk so that it's not distracting. You're welcome to post your questions in the chat box. And then at the end of the talk, there will also be time for you to ask your questions live. So either one of those options is fine. And for people that want to receive SACNASP points for viewing this or for the participation here, please just leave your name and a note about that in the chat. And we will make sure that that's sorted out. Right, I hope that that is everything. Hope I didn't forget anything. Daniel, we look forward to it, so you may go ahead. Thank you very much.
Thank you, Anina, for that nice introduction. And hello, SAAB members. Thank you very much for the opportunity to present my PhD work. And I trust you will find it interesting. I would want to start sharing my story. And this is one of the reasons why upfront I put on these beautiful images of Thesium, because of my past experience with the plant. So I call it my story because it's a narrative of my PhD journey. but I would rather call it a confession to make, because prior to my PhD, I didn't know this cesium, the subject of my PhD. So I had to read books. Can I continue? Okay, so I had to read books, and particularly the Flora Capensis became like a Bible to me, because I practically read it every day, getting to know the plant called Thesium. So from these books, I was able to see that Thesium is worldwide distributed, particularly in the Old World, and by introduction to Southern and Northern America. So, but all these literatures are pointing to the fact that Thesium is highly diversified in Southern Africa, particularly the Great Cape Floristic region. So my story started this way. When I arrived South Africa in Cape Town 5 days after the arrival, Muthama said to me, Daniel, I have a meeting in Hermanus, and I would want you to go to the field to botanize it. So I said, of course, the answer should be a yes, because I didn't want to say I am not ready. So I said to him, yes, we're going. And after that, we went there. He said to me, while he is attending the meeting, we can go into Franz Kloof Nature Reserve to see what is there. So Tammy, postdoc in the lab, and myself went to the reserve. And in the afternoon, we came back, and we were resting, and Muthama joined us. And Muthama asked, and he said, how many Thesium species have you collected? I said to him, there's no Thesium in this reserve. And as usual, Muthama smiled, and he said to me, if you're not tired, let's have another look. So we went into the reserve again. A few meters from the visitor center, we met a huge farm, or a population of Thesium strictum. And after the loop, we came back with 9 clearly different species of Thesium. So my story, it's about starting something that is within my expertise, but the subject of the study was actually not very clear to me, but in the end, I was able to establish the fact that there is high diversity of Thesium in the Cape, particularly the Cape region, and I've witnessed that. So as a result, I was able to look at the general habitat and habits of Thesium. And I saw that some of them are prostrate small shrubs. Some of them are erect, but very short, less than 10 centimeters tall, and they are herbaceous in nature. And some of them, they are actually large shrubs to some extent to a big trees. And so we looked at the flowering time, because sometimes you go to look for plants that flowers at a particular season. We found out that the flowering time for Thesium is between August and January, and a few species flower around April, May. And I read about the knowledge gap initiated by Moa, who was also in Tony's lab. He initiated the aspect of the molecular phylogenetic relationship of Thesium. And then Thesium was segregated into about 5 genera. So looking at the molecular phylogenetic relationship of Thesium based on that study, the study used 2 markers, ITS and TONLF. Although the study was robust, but it was not well resolved, because the use of the markers were limited and the sampling was also not complete. So another study on Thesium to confirm what Moa did and the suggestions by Moa, the study by Nikrent and Garcia in 2015, they used ITS, a single marker, and from their phylogeny, I was able to see that the relationship within and between species were not resolved, forming polytomies, and the sampling was not complete. And you could see that just few handful of the Cape species and the representative of other species from the world. So looking at the gap between the work of Moore and the work of Nikrent, I was able to see that there was a need to sample more of the species and to increase the molecular markers. So with this in mind, we asked whether Theseum is a monophyletic genus. Based on our expanding loci and taxon sampling, we confirmed that the genus Theseum is actually monophyletic relative to the segregate genera. So the collapsing of the 4 segregate genera, Thesidium, Ostromeraceum, Concolella, into Theseum was confirmed by this piece. And each of these clades was confirmed to be monophylogenetically strong and with a strong biogeographic pattern. So from our phylogenetic tree, we're able to see that the biogeography of each clade was supported by the molecular work, and the subgenus Hagnothecium, for instance, which we recognize as a subgenus, was endemic to the GCFR, the Greater Cape Floristic Region. The subgenus, which was the type subgenus, were mostly of the origin of Eurasian species. Discothecium was also part of the subtropical Africa. Subgenus Discothecium and subgenus Xylothecium were from that extraction. In these clades of Xylothecium were also included the American species, Theseum tupiensis and Theseum brasiliensis were within the subtropical clade. But the bulk of the Theseum, which we named or we recognized as subgenus Frisia, were of Southern African extraction, particularly the Cape region. Now, putting the current work within the historical background of the classification of Theseum, we were able to see that section Frisia, section Ithethecium, Annulata, Barbata, Penicillata that were previously recognized by the previous workers were collapsed and they were recognized under the subgenus Frisia. And Euthecium, which was also called section Imberbia, and Fimbriata was collapsed under subgenus Theseum, and then Chrysothecium and Subglabra and Discothecium were classified under Xylothecium. And then Thesidium, which was recognized by the Condell as section Hagnothecium, were recognized based on our study under the subgenus Hagnothecium. Now, after this robust work, and after the phylogenetic classification, of course, one of our aims is to be able to have a resolved phylogeny that will allow a piecewise taxonomic revision of the genus, particularly that the South African taxonomic research has prioritized Theseum as in need of revision. So the idea was to have a robust phylogeny that will allow the taxonomic revision of each clade one at a time. So at this point, we're able to look at whether morphological characters allow or support the phylogenetic relationship in Theseum. So for this, we are able to score 42 characters and then optimize 10 of those diagnostic characters on the phylogenetic tree obtained from the previous aspect. So we looked at, I will give example of 10 of them, 5 of them instead of 10. The first one, which I consider very significant or very important for recognition of Theseum, is the aspect of the presence of trichomes, or as others researchers call it, hairs, or others bears, on the apical aspect of the flower. So for this study, we call them trichomes, and we scored for presence, absence of this character. So we mapped it and optimized it on the tree, and we see that the presence of trichomes at the apical parts of the flower is a plesiomorphic character for some species in subgenus Frisia and some species in the subtropical African species, Silithium. The lack of trichomes on the flower is an ancestral character for the genus. And then we looked at the type of inflorescences. We looked at the various types, and we categorized them into 5. And we saw that the solitary flower, the cymos flower, and the rhizome are derived characters for the genus. Then the spikelet flowers are ancestral for Theseum. Then we looked at flower merosity, that is the number of lobes on the flowers, and we scored for either 4, 5, and above. And we consider the 4 merose character for subgenus Hagnothecium as synapomorphy for that clade. But on the average, or mostly, the 5 merose character is the commonest amongst Theseum species and is considered as a derived character. Then we looked at a character which is autapomorphy for one species, which is Theseum euphybioides. Theseum euphybioides have this character as autapomorphy because it's clasping the stem, which is unique to this species. We looked at the relationship between the stigma and the anther, which we also mapped, and the tropical species called Silithium have their anthers rising above the stigmas. And we consider this character as apomorphy for this clade. So of the 10 characters we mapped, I'm giving us only 5, and these other characters can be read in one of my best papers, and I think is the best ever, that was published in Taxon. So for the details of the other characters, you can refer to this paper. Now, like I said, the aim of the phylogenetic study was to see whether we can have an opportunity to revise each clade. And we took the first clade at the base of the tree, which is subgenus Hagnothecium. We took this clade for taxonomic revision because, one, it is Cape endemics. All species in this clades are Cape endemics, and there were high species complexes with a lot of taxonomic confusion in this clade. So we decided to take this piece and revised the clade. And the synapomorphies observed for these clades are that, one, they are dioecious, having separate plants, male and female plants, and the male and the female plants are heteromorphic. So you hardly differentiate, you hardly recognize the two sexes as separate, as the same species. But one way to know that these two are the same species is that they are cohabitate. They are always found together in the same habitat. So they are heteromorphic, but always together. You know, husband and wife, they said they move together. So that's one of the ways to get to know that they are the same species. And the flowers are exclusively fourmerous. The flower lobes are always 4 in number, not 5 like the other species in Theseum. And then the perianth lobe inside out are green. Like you see here, the inside and out are green in color, which is unique to these clades. So we consider those 4 characters as synapomorphies for this clade. So we collected both qualitative and quantitative characters and subjected them to univariate analysis to see where there's a discontinuity in characters. And bracteal length, for example, have presented it, has been used as a diagnostic character for these clades, and particularly the brittle species have shorter bracteal length compared to those species like Theseum minus and Theseum fruticulosum. The male leaf length and the female leaf length is another character that is also diagnostic to this within species of this clade. And other characters like the plant height, the stem diameter, the flower length are a continuum for those species. And then if you come to the corolla breadth, the diameter of the corolla or the perianth, you will see also there are some outliers in those characters. So we perform this univariate analysis to see whether we can see a break within the species, break in the characters of the species. And to be able to have a robust analysis, we also performed the principal coordinate analysis on both the quantitative and the qualitative characters. PCOA because it allows the use of both discrete and continuous characters. So based on this PCOA, we are able to recognize 8 species as unique and 3 species were synonymized, were collapsed into other species. So for instance, Theseum thombaji, Theseum fragile were collapsed into one, and based on the rule of priority, Theseum fragile was recognized, synonymizing Theseum thombaji. Theseum minus, Theseum hirtum, Theseum fruticulosum, and longicoli were recognized as distinct species. But Theseum exocarpoides and Theseum microcarpum were also collapsed into one because the cluster analysis did not give us a clear cluster of them to be separate species. Therefore, 8 species were recognized based on this analysis. And then in the course of our exploration, we described 1 species as new to science. And this was also published in this paper. So for details of the revision of the subgenus Hagnethisium, one could refer to this paper. And within our phylogeny again, we were able to see that some species were outside the Hagnethisium section, but we needed to describe them because of the need in terms of knowing that it will be difficult to revise the whole genus at a time, and the need to describe these species before they go into extinction, and given that their habitat are vulnerable. So we decided to describe 6 species that are endemic to the Greater Cape floristic region. And this description, we used the approach of the several lines of evidence, using the morphology, using the molecular phylogeny, distribution range, and the ecology of those species. And based on our study, we're able to describe a new species. We called it Theseum aspomantanum. Aspomantanum because we collected the type locality is at Skuverberg. Let's use that because it's called Rough Mountain in Afrikaans, and that has been transcribed to Aspomantanum. The second one was Theseum demagii, which was named after my wife for her sacrifice during this work. Thysium neoprostratum, because it's very similar to prostratum, to Thysium prostratum. Thysium sour, named after my mentor in Nigeria, Professor Fatima B.J. Sawa. Thysium nigro perianthum, because the perianth are blackish in nature, so, and unique to this clade, so named Thysium nigro perianthum. Thysium rhizomatum, because the underground is rhizomtous. That is, it forms rhizomes, which is unique to this plant, and it's erect, but mostly less than 10 centimeters tall. Very short plant, but very nice and good looking. So it was named Thysium rhizomatum. This was named after Professor Charles Stratton, who collected Thysium before my arrival, 10 years before my arrival to Cape Town. So he himself and Muthama collected a lot of Thysium specimens that have saved out a lot of time going back to the field, and we used those resources. So we deemed it fit to name one species after him. So this was also named Thysium statoni. So that was the aspect of the phylogeny and revision. The next question is, what is the applied aspect of this work? Applied because conservation of unique biodiversity is hampered on taxa with ambiguous nomenclature and classification, particularly those with unresolved phylogenetic relationships. So because of that, we looked at what are the extinction risk patterns in Thysium, particularly those that are of the GCFR clade. We tested the hypothesis that range size, ecological specialization, and extinction are phylogenetically structured such that climate change can prune the diversity of the Thysium, the GCFR Thysium. And for this, we model the past and the future ranges of Thysium based on the present day locality point of species and their environmental conditions using Maxent model. And based on this, we are able to see that pulling the best 9 bioclimatic variables and 6 soil variables and 1 elevation character into this analysis, we're able to see that there was a strong positive correlation between species range extent and those environmental variables. And from this, you can see that each of these dots represents a species, and this gives us, the blue line gives us the best line linear fit for each species, and the gray band shows us the level of confidence of this analysis. And you can see that each of them showed a positive correlation, except for Bio 5, which is the maximum temperature of the warmest month, and soil potassium, soil nitrogen, which did not show any relationship of the species range and environmental variables. We looked at the species distribution modeling, trying to look at it in the context of maps. Here, each of the species were modeled. We modeled 101 species, but I'll give us here 4 scenarios. The first scenario is for Thysium hispidulum, which shows that the past, which can be traced to 22,000 years ago, the last glacial maxima, showed that there was a broader range extent, suitable habitat for Thysium hispidulum. And then to the contemporary time, there was reduction or contraction of the suitability of the habitat. Into the future, there's drastic decrease in a suitable habitat for this species. And these maps can be represented by the bar charts below. And you will see that the bar chart clearly showed that there was more suitable habitat for this species in the last glacial maxima compared to the present. And into the future, this species would have a strong extinction threats. The B1, which is Thysium strictum. Thysium strictum is widespread species of Thysium across South Africa. And so I present here because to see how this species can be threatened as well. The pattern is almost the same between the past and the present. The frequency of the cells are almost the same, but into the future, this can also, the suitable habitat can also reduce. And this could be as a result of the threats of climate change on this species. The C aspect, which is Thysium namoquense, is another scenario with limited suitable habitat in the past, with increase in the suitable habitat as the environment gets warmer and drier. As it goes into 2080, the suitable habitat will continue to expand because this Thysium, by the analysis, it suggests that this Thysium is a lover of drier condition, warmer environment. So it is expected that Thysium namoquense would expand into the future. So it may not face the extinction threat imposed by climate change. Another scenario is Thysium spinosum. In this scenario, most suitable habitat for this species were around the eastern coast. As the condition gets warmer into the present, there was a shift in the suitability of the habitat into the western coast. And then into the future, though the habitat will not change, but there will be a shrink and the cell will reduce, the cell frequency of this habitat would also reduce into 2080. So these are the four scenarios that the model predicted for the species occurrence of Thysium species of the GCFR. By and large, out of the 101 species we modeled, 71 species were predicted to have had broad range size during the last glacial maximum. 27 species, which represent 17%, recorded range reduction historically to present. 45 species will potentially can I change this place? Okay. 75 species will potentially expand their range breadth by 2080. 51 species, representing 50 species, will potentially reduce their ranges by 2080. 10 ecological specialists, interesting enough, represented by 8.5%, although have had a range reduction from the last glacial maxima to now, but were predicted to persist even in the face of climate change into the future. Mapping these characters, species range extent, ecological specialization, and extinction risk on the maximum credibility tree obtained from the previous chapter. And then looking at the phylogenetic signals, we observed that there was lack of phylogenetic structure of these characters of species range and ecological specialization and extinction risk on Thysium. And if you look at it, you will see that those tips with reddish, yellowish lines represent species with small range sizes, and they are randomly scattered on the tree, showing lack of phylogenetic signal for that character. And same can be said on the ecological specialization, seeing that the reddish tip here and few of the yellowish ones represent species that are specialists and the blue ones are generalists, and they are scattered on the phylogenetic tree. And any of the phylogenetic pattern observed in the GCFR Thesium clade is not actually a function of structured variation, rather is a selection of convergent evolution. So this indicates that range size, ecological specialization, and extinction risk are evolutionarily random and therefore should have a negligible impact on the phylogenetic diversity of the Greater Cape Phoretic Region Thesium. This was also sent for publication with the Conservation Science and Practice, a journal of the Society for Conservation Biology, and it has been accepted with revision. Not published yet, but we look forward to seeing this published. To conclude the talk, I have to thank these two wonderful people who were amazing during my research. Professor Muthama, they took me around South Africa looking for Thesium. They supervised this work, and it's with their help that I was able to do this work. So Professor Muthama and Professor Anthony Van Doom, I am grateful to them. And I also want to acknowledge the science faculty, UCT, for fellowship granted during this work. They provided fellowship for 3 years during this work. And thanks to Gombe State University for allowing me to pick up this position at UCT. AIPT came in lately to augment the aspect of DNA extraction and the overbear conservation trust that was always there for us, allowing us into their property to take Thesium materials. NRF sponsored the aspect of laboratory work. So the DNA, the molecular work was done under that funding. So thank you so much for the brief presentation on my work. And that is it.
Thank you so much, Daniel. That is excellent.
Thank you.
Let's get this right. Thank you. I'm going to keep an eye on the chat for any questions. The audience is welcome to leave questions in the chat function there, or just raise your hand and then you can ask your question as well. Daniel, I just wanted to ask you quickly about that ecological specialization and the extinction risk that you showed there. How did you measure that, or what kind of traits were included in ecological specialization?
So what we did was we extracted environmental variables for the locality points of our species, and then use the mean, the standard deviation of the means of those points. So for example, if we had taken the soil variables, the nitrogen, potassium, EC values and so forth of the locality point, and we got the mean and the standard deviation of those environmental variables. And then we subjected the standard deviations to principal component analysis. So the PC1 and the PC2, which were the major contributors of that analysis, we took the first axis, the PC1 values, as our index of specialization. So for instance, if you have a species with a small PC standard deviation, it suggests to you that it's a specialist. Species with a bigger standard deviation suggests that it's a generalist. Is that better?
Yes, yes.
So,
no, that makes sense.
So that became the index of specialization. The smaller the index, you are a specialist. The broader the index, you are a generalist.
So it was mainly abiotic factors influencing their ecology that was considered there.
Yes. So
Okay.
abiotic factors to determine their niche breadth, for example.
Yes.
Yeah.
Okay. Glynis has a question, and then after that, Kenneth. Glynis, please go ahead.
Thanks, Anina. Thank you, Daniel. That was really interesting and very well done once again on that challenging group that you were working with. So I was quite interested in your decision to combine, to synonymize some species based on your principal coordinates analysis. And I just wondered if that was the only basis on which you made that decision. And what sort of species concepts were you applying? Was it purely morphological, or did you look at ecology? Did you look at DNA? Anything else?
Yeah, so our decision for the taxonomic revision of that clade was based on several lines of evidence.
Okay.
So morphology, of course, was the first. And so we took the qualitative and the quantitative characters, and that we used for the PCOA. Then we looked at ecology. Is there overlap of habitats? That was also considered. And for example, if it was Thysium fragile, which was mostly found on the coastal sand, was also scored as coastal species. And other ecological characters like the habitat was also scored. And these were pulled into the Gawa distance in PCOA. And that gave it a strong, distinct clusters that you saw in the PCOA. So we used morphology, we used ecology, particularly the distribution, and supported by our molecular data. Of course, we were informed by the species relationship in the phylogeny, and then looked at the characters of these plants on the herbarium specimen. This together we pulled. So we discovered that the morphology supported our claims in the phylogeny. So it was a kind of several lines of evidence to support our decision to synonymize those species. Is that better? Explain your question.
Yes, it looks like Glynis's internet is hanging now, but I think she got that your answer. Thank you very much for that, and thank you, Glynis, for the question. Kenneth, do you want to unmute and ask your question?
Afternoon, Daniel, Anina, can you hear me?
Yes, I can hear you.
Great. Thanks, Daniel, for a very interesting talk. My question relates to, and this is purely ignorance from my side, but potential biotic aspects that might affect extinction risk in Thesium. And the obvious one that comes to my mind is host specificity. So purely from a place of ignorance, is there actually a degree of host specificity for different Thesium species? Do you find that they parasitize different host taxa, host genera, host families, or things like that?
Yeah, so several literatures have stated that Thesium is semi-parasitic in nature. And I read some articles on that. And for my curiosity, anytime I was in the field, I will dig up to see who is the host of Thesium. And I dig and get tired. And since that is outside the scope of my study, I did not delve too much into it. But the literatures are saying that Thesium is not actually exclusively a parasitic plant. So it parasitizes a plant at early stage of its life cycle and then becomes independent in the later age. So our scope was not into the parasitism aspect, and we stopped at the systematics aspect, but we could not establish, based on our field experience, we could not establish a specific host for Thesium. Some Thesium, you find them in an open space. Difficult to say where is it coming from, how how does it parasitize anyone? But probably in the roots. Of course, it's a root parasite. So we could not establish the aspect of host specificity for Theseum. So I think that is what I can say, but the literatures are suggesting that is a root hemiparasite.
Thanks very much, Daniel.
You're welcome.
Thank you. I'm looking out for other questions. I haven't seen any in the chat yet. I hope I'm not missing anything. But Natty has a question.
Yeah.
Please go ahead.
Thank you very much. Can you hear me, Daniel?
Yes, I can hear you, Natty.
Yeah, thank you very much. First of all, congratulations on a
Thank
lovely
you.
presentation. I learned a lot about Theseum that I never knew about at all. Thank you for that. My question to you is rather simple. Are you aware of any chemical work that was done on the Theseum? Because I'm known many times before one decides whether two species are in fact conspecific, that they sometimes use chemical markers in order to try and assist you with the morphological markers. In other words, using a chemotaxonomic sort of classification in addition to what you do on the morphological side. Just curiosity.
Yeah, so for now, there is no study on the chemistry aspect, much that I know. I know of a lab in Pretoria. Natasha and Marianne are working on Theseum, especially the Xylothecium clade, and our works are complementary. And they have looked at some aspect of ethnobotanical aspect of Theseum. But even on their part, there was no work that I know on the chemistry aspect.
Mm.
And I think Theseum is edible for plants, for animals. And so I did not see any potential in the use of chemistry for the taxonomy. So, and probably because I did not consider that line of evidence, but it's worth trying. So maybe Natasha and Marianne will take up with that because they are into the ethnobotanical uses of Theseum. But for the taxonomy, we did not explore that path.
Natasha, please go ahead with your question.
Hi, Daniel. Thank you very much for such a wonderful talk. I'm just tying in with Natty's question. We actually have, Marianne Leroux and I actually have worked on the chemosystematics. Our paper is currently in review. And it's quite interesting. We found that we looked at the phenolic compounds of about 50 species, Theseum species, and we found that they are very specific for each species, which rather surprised us, as phenolic compounds, I think, can sometimes be very general. And we found that some species with different phenolic markers can actually be distinguished. So there were species that we, for instance, thought were a single species, and then we found two completely different phenolic profiles. So this is just a preliminary study that you can look out for as soon as it's published. We have submitted to the Diversity Journal, but I think there might be some very interesting results if we look at that aspect further. Unfortunately, we didn't find any phylogenetic signals with the chemophonetics. Yeah, so just to tie in with that.
Yeah, so the fact that Theseum is edible to animals is suggestive of not much would be gotten from the aspect of the phytochemistry, probably the proximate analysis but the phytochemistry, phenolics, terpenes, alkaloids, and things like that, I see no potential of using it, particularly if you are using that character for taxonomy. Yeah.
Thank you very much for that. We still have some time for questions if there is anybody. I was wondering about, you mentioned that only one of the species had rhizomes. So that made me wonder about the fire regeneration strategies in these thesioms. Do you know how diverse it is and whether it's scattered across the phylogeny or not?
For now, we did not score the aspect of rhizome. Just that for curiosity, we were able to dig up for that Thesium rhizomatum, and we saw a huge rhizomes underground, which was unique to that species. So we decided to look into other species, whether we can see those characters, but we didn't find anyone with that character. So it's unique to that species. And if there's more to that, maybe it's for another study, but the presence of rhizome was unique to Thesium rhizomatum. And the aspect of resprouting after fire is also a potential to explore in Thesium, because some Thesium thrive better when or after fire. So it's a path worth exploring, but for now, we could only identify the presence of rhizome in Thesium rhizomatum.
Interesting.
Yeah, thank you.
Great. I do not see any questions in the chat here. Is there anybody else that has any questions? Yes, Kenneth comments that there's lots to explore underground in these thesioms. So hopefully we can get some curious students that's willing to dig a bit.
Yeah.
Yes, Mfama, please go ahead.
Thank you, Daniel, for that interesting piece of work. Where to from here? What are the gaps that need to be filled? I'm sure you did not do everything, Daniel, did you?
I didn't. Yes, even though we claim that our phylogenetic tree is robust and well resolved, we still find difficulties in some clades. For example, the Hagnothis, the Frisia, let me share. The subgenus Frisia remains problematic to us here. And Hill recognized sections, barbata, annulata, Frisia in these clades of subgenus Frisia. Species boundaries are still complex in this clade. So there's a need for a taxonomic revision in this clade. If we want to pull out Thesium as a priority in South Africa, then there's a need for a revision of this clade. Happy enough, Natasha and Co have resolved this. I think they have taken good care of this. And together we will explore this path. And we suspect hybridization and other factors that could lead to this unresolved relationship within subgenus Frisia. So in Thesium, we can still see potential work in the bulk of subgenus Frisia for taxonomy and for hybridization studies. So that is our target, to be able to pull out Thesium from the priority taxa. We need to revise subgenus Frisia, and it remains a problematic clade. I think Natasha can tell us more about the problems she encountered with this section.
Thank you very much.
Yes. Sorry. Yeah, just to link in with what Daniel is saying, it is quite problematic. that specific subgenus, but I think working together, we will tackle it and hopefully make everyone's lives that needs to identify Theseum much easier.
Yeah.
Thank you. Glynis, please ask your question, and then thereafter Kenneth.
Thanks, Anina. Just seeing this phylogeny again reminded me that I'd wanted to ask about that single South American species in the middle of that tropical African clade there.
Yes.
Any comment on how you think that got to South America?
That Theseum tupuense and Theseum brasiliensis, which is not shown here, they are from America, introduced to America. And based on, it was called genus Ostromericium. And so it was one of the segregate genera called Ostromericium, but the phylogeny suggests that it's actually Theseum. So it's one of those segregate genera.
So when you say introduced to South America, it's not naturally occurring there?
Yes, it's not naturally occurring there. So if you look at our map here, okay, here you see this purplish part as introduced species. So it's Theseum tupuense and Theseum brasiliensis in Brazil. So it's not natural, it's introduced species. So probably from tropical Africa, the origin.
Okay, thanks.
You're welcome.
It looks like Kenneth had the same question there.
Oh, yeah.
Muthama, yes.
I would like to comment on that odd distribution of Tepuenses to South America. And if I'm not wrong, it's occupying substrate, which is also nutrient poor, like in the Cape. And it's an odd biogeographic linkage, but it's not unique to Theseum only. For those who are familiar with Paomites, Prionium serratum, the sister taxon, Thunia, is sitting also in the Guyana shield. And so, while I can imagine that Theseum might have used the tropical African route to disperse to South America, and that pattern is shared by giant Synesios and giant Lobidias, which have got a tropical Africa, West Africa, and South American link. For Prionium, even puzzles me more that it's from the Cape directly to the Guyana shield. But more questions than answers for some of these groups. Thanks.
Thank you very much, both of you, for those answers. Are there any last questions? I don't think so. Time is just about up as well. So just lastly, thank you again, Daniel. That was a really great presentation and a really great contribution to that group. And I'm sure all of us will have a new search image for Theseums when we're out in the field now and be looking out for them. And thank you everybody that joined in. It was great. Our next webinar will be in August, I think 17th August. So please look out for that. And we hope to see you all there. And this presentation recording will be available on the website of SAAB. So you can go and access it there if you need to. But again, thank you very much.
Thank you for having me.
It's been great, thanks. And thank you everybody. Hope everybody has a great evening.